Colin Semple, MRC Human Genetics Unit, Edinburgh. The evolution of homologous recombination repair deficiency in high-grade serous ovarian cancer. High-grade serous ovarian cancer is a deadly disease with poor survival rates and is also comparatively poorly studied with whole genome sequencing. The largest previous study involved 80 primary tumours and found abundant structural variation with somewhat old-fashioned algorithms. It also found frequent mutational signatures of HR repair deficiency and frequent mutations within the coding sequences of the BRCA1 and 2 genes. BRCA1 and BRCA2, of course, have well-established roles in DNA repair and underlie the effectiveness of PARP inhibitors due to synthetic lethality between repair pathways. But at the moment, only a modest proportion of patients with disruptive short variants in BRCA1 and 2 get PARP inhibitors. We've examined the frequency of structural variants at these genes to uncover new patients who may benefit from these promising new treatments. We've assembled the largest combined cohort of whole genome sequence tumours to date, with matched RNA-seq for most samples, and uniform processing and analysis of all data. We've also used the most recent algorithms for structural variant detection and an algorithm called HR Detect, recently developed to infer the presence of homologous repair deficiency. So we found many samples with large structural variants at the BRCA1 locus, which you can see here, including duplications, inversions, but dominated also by deletions. And we found that these large deletions, which are many megabases in size, are also associated with a decline in the expression of the BRCA1 gene. We see a similar pattern at the BRCA2 locus on a different chromosome, where we have very large deletions which have a similar impact, decreasing expression. So we've established the presence of these classes of new large variants, but do these alterations result in an HR deficiency phenotype in tumours? To find this out, we've used HR Detect, which is the current gold standard prediction algorithm, integrating a variety of genome-wide mutation patterns that can indicate repair deficiency. This graph shows each tumour along the x-axis from a variety of our tumours and the HR deficiency score on the y-axis. And those above the red dashed line are predicted to be HR deficient. We can immediately see that many samples with HR deficiency have the traditionally found damaging short variants in BRCA1 and 2. But many others do not, and instead carry structural variants. And in particular, they carry these large multi-megabase deletions that span the entirety of either the BRCA1 or BRCA2 genes. And these large deletions independently affect a proportion of patients, in addition to those affected by short variants within the BRCA1 and 2 coding sequences. So these are large, complex, rearranged genomes, and these large deletions at BRCA1 and 2 often co-occur with other mutations. But this graph shows that double deletions, that is, deletions affecting both BRCA1 and BRCA2, or deletions that occur in combination with damaging short variants, are associated with strong HR deficiency. We also see effects for short variants, of course, as expected, and also for BRCA1 promoter methylation and for certain inversions. All high-grade serous ovarian patients receive platinum-based chemotherapy, and that's known to be more effective in patients with HR deficiency in their tumours. And correspondingly, we do see that in our data, where we see that repair deficiency from any source promotes survival. But importantly, we also see that repair-deficient tumours which lack the short, traditionally found variants in BRCA1 and 2 are also associated with better survival across the cohorts, implying that these large deletions have an equivalent effect on the function of the BRCA1 and 2 genes. This raises an additional question, which is how often is BRCA1 and 2 function disrupted in other cancers? And for this we've exploited the recent ISCB pan-cancer analysis of whole genome sequence, which covers many different tumour types. And in here we see substantial frequencies of large multi-megabase deletions encompassing BRCA1 or BRCA2. And in each case where we have power to estimate it, we see an impact on expression such that deletion-carrying tumours have lower expression of the genes affected. So in conclusion, we've found frequent multi-megabase deletions encompassing the BRCA1 and 2 genes can contribute to homologous repair deficiency independently of short variants. Overall, an additional 16% of patients appear to benefit from PARP inhibitor treatment, in addition to those affected by damaging short variants within the BRCA1 and 2 coding sequences. Impaired BRCA1 and 2 function, arising from deletions or other sources, is also associated with better overall survival for patients. And many other tumour types appear to show similar disruptive deletions at BRCA1 and 2, suggesting that the potential of this kind of homologous repair deficiency extends to other cancers. This is the work primarily of a talented postdoc, Ailith Ewing, in the lab, along with Alison and Graham, who did most of the work. And this is a collaboration with Charlie Gourley's lab, who is an oncologist in a neighbouring institute. And the study is now available on bioRxiv. And the full acknowledgements look like this.